Well, thank you all for coming. I guess that the expectations are high. I wish I won't disappoint you. And well, I'm also assuming that the psychedelics topic is quite hot and it's quite interesting also. But in fact, I'm going to talk about a paper in psychedelics. One of the authors is Robert Carhart Harris. I don't know if some of you are aware of him, but it's like promoting research in psychedelics quite a lot for some years now already. But the idea behind this is the next project of my PhD, the one that I'm starting, I hope, very soon, is about analyzing brain networks using other different techniques than just what is like been using for 15 years already now, which is statistical measures and connectomics. You can even say with a spectral analysis. So this is like, well, I thought sort of also to help me understand this a bit more, because it's not an easy topic to familiarize oneself. And of course there's the interpretation of these things, which makes it very, very complicated, because if connectomics is already very difficult to understand, so basically we can know more or less what global efficiency, path lengths, all this means, but can we actually go beyond the simple math of these and translate it into clinics? There was a very recently there was a very nice piece of opinion. I think it was Nature Medicine, that why is it so hard, even that we've been doing connectomics for so many years, to really apply it to the clinics? And it's because it's very, very abstract. Yes, we have some clustering, we have some communities, but like, what is the real application to clinics? And it's challenging, but I do think that it's worth to keep investigating that. And of course, people who are promoting this want to keep doing it because they think, of course, that it's also valuable. So just to give an overview, what's going to happen is that I'm going to start with what we know. But hopefully most of you follow me, at least to here. Like, I would say that's pretty okay. And then like all of this is to introduce this new technique, new analysis. Okay, this I think I'm going to skip this one because I have not timed the presentation, but I think it will be too much and I have not really prepared the last one. So I will start with why is it doing like this? Okay, well, so this is the normal workflow. I would say there are two specific branches. That's how it works. You start with an MRI, you do a T1 weighted scan, and then you do some diffusion and you get some tractographies there. It should be some kind of dynamic, but for some reason it's not. No. Okay. And then you end up like connecting you parcellate the brain into regions and you count how many, let's say, fibers or streamlines go from one point to another, and you get a graph which is characterized by a full adjacency matrix. And you have another modality, which is like you do the T1 and then you do some bolt analysis with maybe some experiments meanwhile the person is doing it. You get with some signals and you correlate them. You can do many measures, but the principal one, the main one, is to correlate those signals, and you also get a matrix of how different regions in the brain somehow get co-activated together. And you can see that the structure and function look very different just just by looking at them. The nice thing about this is that you end up with graphs that you can also characterize some, let's say, structural diseases, some structural injuries, and also some behavioral. So there's been studies with schizophrenia. I think that this is more or less known for everybody, so I will not spend too much time. And then with these graphs, then the mathematicians and physics bored with particles. Then they started doing some things, and they invented or they designed some connectivity measures to know how the interactions between the different nodes in the network are related together. This is a very famous paper, and you have like lots of studies of this. Like, this paper has been cited over 10,000 times, and there are many books about this, so you can do many things, including longitudinal studies. So you can measure these properties of the networks over the years, and you find some interesting results, and also in schizophrenia. So this is, I think that most of you are familiar with this. It's just some, let's say, graph theory measures, right? I'm not so sure if you're familiar with the idea that these measures, despite calling topology of the graphs, they are basically statistics on the nodes. So you have one node and you count how many links they are, and then you do some fancy statistics with cool names, and then you end up with clustering coefficient, with number of edges, degree, global efficiency. There are many. But I really recommend for anyone doing graph theory to read this, or at least to grasp this paper. It's very recent actually, and it's quite hard in terms of mathematics, but it highlights the idea that these statistics do not come with their flaws. So for example, they put a very nice example here where they say these are clearly super different networks, right? Even though if we rearrange the nodes, because we can do that, they are super different. But the network statistic that we usually use, which is, for example, number of vertices, edges, the strength, the clustering coefficient, they all have the same value. So that is a problem, of course, because we may be in two different, I don't know, I'm just inventing something, but this may be a control case, schizophrenia, blah, and then we get the same characterization of the graph. So that's a problem. And even more, so they go a little bit, they analyze this idea even further, and then you say, okay, let's compute the modularity of lots of networks, and let's also compute different other measures as well. So we use the global clustering coefficient, global efficiency, and they are highly correlated. So if you report the modularity and the clustering coefficient, you're basically reporting the same thing. So it doesn't add any value, right? To the study, for example. In some cases, it's quite evident. If you compute, for example, the small worldness, which is the clustering divided the path length, it's somehow obvious that they will be correlated because one is computed with the other measure. But in some other cases, it's not so obvious, for example, in modularity. Even more, there's a problem, especially in functional networks, when you build the correlation that I told before, but also be it instructional networks, that you don't know what is noise and what is not noise. So you usually say, okay, let's cut correlations below 0.1 are just noise, so let's drop them. But where do you put the threshold, right? It's a big question. There are some, even I don't think there is a big consensus on that yet. Even more, so okay, I threshold at 0.1 and you threshold at 0.2. Let's do our study of graph theory measures, and there will be obviously a difference because the network is completely different. Right? And just as a thing, we also did that, and you can see that the modularity at different thresholds, it's like completely different, and also the entropy of the network. So indeed, thresholding the graph is a complicated thing to do, and it can really affect your results. That is, I believe, and here raises a question, like what is the weak connection on a functional network? Is it really a correlation of 0.01? Is it real? Is it not? Why are we thresholding it? What's like? It's a bit of a dark box. Why we threshold and how we do it. Okay, and this is where topological data analysis, I believe, has its major strength. We believe, I guess, that you too. Right? But we need to start like with topology on surfaces because topology is very abstract, even though it deals with very common objects such like the Earth and the donuts, right? So hence the title. So I would ask, well, this is a shame because this was supposed to be sequential, but you see the whole picture now. What do Earth and donuts have in common? It's basically nothing, right? So let's do the mathematical abstraction of this. We'll just draw a sphere and let's draw a donut. And you could also see that the difference is quite big. But if I would ask why are they so different, I don't know if I would get the correct answer thinking on topological terms. I would say that most of you understand that there is a hole in the donut, right? But this hole actually is a very strange hole, so we're not talking about this hole when we talk about topology. We're talking about, in a way, how can we encircle the hole so that we cannot compress anything to one point. So there, the torus has actually two holes, because you can draw a circle here and you cannot compress it, and you can also draw a circle there and you cannot compress it, so it has two holes, while the sphere doesn't have any. You can draw any line, close the line, and it will compress into one point. But even more, how many holes has a sphere? I believe that all of you, or most of you, will say 0 holes, right? But it's not true. It has one two-dimensional hole, because if you draw a surface and you collapse it into one point, you can't do it without leaving the sphere. So the void, the cavity inside the sphere, is also a topological hole, right? And the same happens with the donut, with the torus, with the donut. right? You will say, why am I talking about this? What's going to be this important for? So hopefully you will believe me that this will be useful. The next question is, we have a cup of coffee there. What does this thing resemble? It is actually a torus, because it has the same number of holes as the torus. You can see the video is quite there. The next question, of course, is how do we measure how two things are equal or how two things are not equal? So then there's the Euler characteristic, which is like this sum, the number of 0D holes, the number of 1D holes. So you start, and you end up that indeed the cup of coffee and the torus have different Euler characteristics, so they are essentially the same topological object, but not the sphere, which has an Euler characteristic of 2. So this is in a way what we call homology. I do like math, but I believe that I don't want to bore you too much. So the concept that I just described is homology, which is an algebraic invariant converting local geometry into a space of global descriptor. But in a way it's like, okay, we have a cow, but we can understand it as a sphere, and that's it, right? So we try to apply, yeah, we are very, very picky, then the cow has some sort of holes as well. But the important thing is that we can actually map two very different things together, and that's what we want to do with graphs as well, right? And we hope that this will provide something that statistical connectomics cannot see, right? That's why people do it, and they found different things. So now we move into discrete spaces of graphs, but we cannot do that super fast, because we need to define what is a hole in a graph, right? So we need to, I said, okay, we will look at holes, but what is a hole in a graph? But as usually happens in math, usually you have 30 pages before what comes in the hole. So we have to start with a clique. A clique is super simple. It's a connected graph. So we have one dot, that's a one clique, that's a two clique, three clique, okay, triangles. So it's a graph that is completely connected between their nodes. Okay? Now the idea is that we understand now a graph as a set of these cliques. So we're not looking at just all the connections, but we say that the graph is, for example, 32 nodes, 52 edges, I don't know, 23 triangles, and so on and so on and so on. And we're actually looking at this. So to compute any topology in this graph, we need to know how many of these substructures there are. Now the holes. The hole, you have to keep in mind the example of the donut is a cycle, right? It's a cycle of what? Of some, let's say, structure or object with the same dimension as the hole itself. So 0 holes in a graph are the connected components, because you can, in a way, compact them as they are. One-dimensional holes are cycles of edges. This is where it becomes tricky. Two-dimensional holes are cycles of triangles. Now here the mind plays a nice situation. We, I would say, intuitively think this of a three-dimensional structure, right? But you should not look at this like that. You have this triangle here, then this triangle here, then this triangle here, and this one here, and this is the cycle we're interested. Because if you imagine this as a cavity, then good luck with this one, right? But if you actually look at the 4 cliques, so these 4 dots here are completely connected, these 4 dots here are completely connected, so then you see the cycle. And these are holes in a graph. Now the interesting thing in topology is that what happens when you, let's say, break the hole. This is what they say, that this is the feature that we're interested in the network, right? So what happens, because they say that this is a significant change, like this is clearly not the same as this one, this small network. So if you, for example, have some piece of information that wants to get here, well, bad example, from here and wants to get here, you have to do all this. But in this one, you just go straight. So this is an interesting topological feature that they want to look at, right? But of course, how do they do it? So what they do is they build this persistent homology, okay? Which is like, we have this network, and we are going to change it gradually, right, until we get to the original network, and we are going to look at the properties of the network at each step. For example, to make it easier, if we start with this let's say this network, this is the original network, right? But in the beginning, since all these networks are weighted, we might have some connections that are stronger and some connections that are weaker. And in the original set, in everything that's been done for years, you just drop the weak connections. But here, you keep all the connections and you see how is the topology changing or not changing. That's why persistent, not changing, across different values of the weight. So that's what you analyze, and you hope that you find something. Hopefully, like until here, everybody's following, or some questions. And now is the case study. The case study is, okay, let's do this. Actually, persistent homology has been done for many, many years, in network theory and in surfaces. But so there are many ways to build and to compute this persistent homology, these small perturbations of the networks, and analyzing the topology of these networks. But this guy actually, which like we hope he will help us, Giovanni Petri, and I guess all these collaborators, designed a new way of doing that works really, really well with networks, and it's the weight filtration process, let's say call it, right? And they define these three concepts basically. So we have the same example that I said before. We have this network. Let's imagine that it's some functional network or any network which is weighted, right? This figure is quite illustrative in a way. And we start with, let's drop all the connections below 0. So, in a way, like we start bottom up. And you analyze, you put the threshold so that connections below this threshold are dropped. And then you have only the strongest connections, which is the first network. And you analyze the topology of this network. You compute whatever you want to compute. I believe you can also compute statistical measures of that, but I'm not sure if it makes sense. Then you start, like you go to the next threshold. You go to the next, in a way you build this object, let's say, that is called the persistent homology, which is a mathematical object. And you analyze, in this paper they analyzed 1D holes, but you can do everything you want in here. You can analyze 4D holes, like good luck with that. So for example, let's look at hopefully with what I explained before, hopefully everyone sees that this red thing is a 1D hole because it's encircled by let's say 2 clicks. But this hole has appeared only at the second step of the filtration process. So there's a change here, right? The topology of these two networks is completely different. There are holes in the second one, they don't appear in the first one. And what they do is like, okay, let's compute at what stage of the filtration this hole appears. And they say in the second step. So let's put this. The hole is born in the second step of the filtration process. You go to the next one, and basically this hole has disappeared. So it dies at the third one. And that is, you compute the difference and you say this is the persistence of this particular hole. And you do that with a whole network. I believe you can imagine, yes, no? I believe you can imagine that this is really computationally intensive. So most of you can actually, 1D holes are computed not easily, but they are computed, but as long as to high dimensional structure, this is really complicated actually. Computationally intensive. So they do that, and they do that for the whole network and for the whole group of studies, but they are interested in the persistence. So how strong are the holes that appear in the network and disappear at the same time? Now I think I have some reservation when it comes to interpreting these things, because what does it mean to filtrate and to keep all the information of the filtration? I don't know what it means. I've thought about it. I haven't really come up with the interpretation of that. Maybe I will ask. So yeah, so now you plot the persistence basically. So you compute how many, along the filtration, you compute how many are burned, how many are dead, and you study the distribution. And for example, how many holes do they remain? 0D holes, you can also do with 1D holes. And basically it's just plotting each hole in the graph has 2 coordinates. It's birth and it's dead. And you scatter this, and you can see like the sequential process of how the topology of this graph is changing over the filtration. You can say over time, but over the filtration. Nice thing is that this is called the phase transition, so it's like water to gas, for example, because the topology is significantly different. But of course you have to interpret what does it mean that a graph has 1D holes or a graph has 2D holes. So that's why it's super complicated to interpret these things in a biological, psychological way, right? Especially if you don't benchmark anything against what is known, which is statistical connectomics. But it's still maybe a worth idea to know.
Why do you have access in fractions?
This one?
No, X and yeah.
So basically, if we look at the red hole, it's born in the second step of the filtration, right?
Yeah.
And it dies in the third one.
Yeah, yeah.
So you put this as a coordinate.
Yeah, I get it, but the scale is in fractions.
Because probably if you go, this could also be percentage. Like if you have the strongest, if it's functional network, the strongest hole edge will be 1, the weaker will be 0. So this, for example, it can be the threshold. But in weighted, in structural networks where you have from 0 to... Yeah, exactly. Yeah. And you plot these things and then you get some nice scatter plots, which means like at what point the topology is changing significantly. Again, what does it mean that the topology is changing in a weight, as long as you changes the weight of the network, right? That's, I think, the difficult process here. And now what they did is like, okay, let's have psychedelics in the middle because psychedelics are cool and it's something maybe worth to look at. Nice thing is that I believe that they say that not much can be seen when you use just statistical and connectomics alone. But if you do this persistence study, then interesting things happen, right? In this paper, they restrict themselves to analyzing 1D holes. This was in 2014, so 8 years ago. But now some people are starting to analyze the 2D holes. I have not seen 3D holes because it's, like I said, super complicated to compute. So they study this persistence between, so the study was like they have healthy subjects and they have subjects injected with psilocybin, which is a hallucinogen or a psychedelic that makes you hallucinate or even to have synesthesia. Synesthesia is like you mix colors with, you understand colors in a different way. And they study the persistence, right? And they say that this is just to show you that this indeed can give at least differences in groups that cannot be seen using connectomics. So what they did is like, let's study this birth diagram, this persistence diagram, and we see that clearly in the psilocybin condition, you have a lot more births like up in the filtration scheme. So you have more weights that are stronger because they get they are born high in the filtration process.
What is the brain signal that they are using?
fMRI. Yeah, fMRI, task rest. Resting state, sorry. Task rest doesn't make sense. Resting state.
What's the procedure?
The fMRI.
They got the psilocybin and they enter in after half an hour. And
I don't remember the times, but I guess long enough so that the psilocybin takes effect. I mean, Carhart Harris is like quite well known in this, so probably they waited and they...
okay.
Yeah.
Okay.
Yeah, well, I think that he's very famous in the psychedelics world. And they studied the persistent distribution of this, of these 1D holes. Remember that we're studying only 1D holes, so cycles in the network. Cycles in the network means that there are not really a lot of direct connections in a cycle. So the information from to one extreme to the other has to go through the whole network, right? So we're studying this. And what we see is that in the psilocybin condition, holes are a lot less persistent. So it means that a hole appears and it gets closed rapidly, right? So don't get too eager on that because it's not, the interpretation is not as simple that it contributes. Okay, this is a measure, a graph measure that they invent, and again they hope to find something, and they do find something, because if you see this one, and hopefully you can also see this one as well. What they say, the persistence homology scaffold, the frequency homology scaffold, so that means the WF, it's only counting the participation of each weight to the holes in the network. And they find that exactly all the weights contribute the same. So there are basically the same number of holes in the psilocybin and in the control, but the persistence of those holes is actually it's significantly bigger in the psilocybin. So again, it's like we have more holes in the altered state of conscious, and they find the same thing here. This is just a scatter plot of those two, and you find that for the same number of holes that specific edge is contributing, the persistence of the holes is a lot bigger. So they are, let's say, more robust. So holes in the psilocybin are more robust than in the control condition. Again, what does this mean, right? I find it interesting because I sort of like all these mathematical tools, but what does this mean? I believe that maybe some of you are like saying, okay, but why should I do this? So believe me that this seems the easiest slide on the presentation, but it was the hardest one, because the same questions that you have about what is this, I have the same. You can analyze these results as this is what we get from the math, but we would expect that this somehow sheds light to something that we can't see using like normal clustering coefficient. And in a way, one thing that I miss in this study is that they don't benchmark against null models, so they don't randomize the networks and compute the same thing, so you don't really know what this means when a benchmark, and they don't really use also the connectomics. So they don't try to use, or they don't explain very well, like, hey, we did the same thing, we compared the two networks with the clustering coefficient, with the global efficiency, and what did we find or what we did not find. Because in a way, I would think that these really much more complicated tools are kind of useless if they don't tell us something that we can't see with the others, right? So that's one thing that I find that I miss in this study. But overall, I think that it's a cool one. Now, I've summarized these 3 points. Basically, the topological data analysis looks at the contribution of mesoscopic structures, which is the holes, because we are not looking at the nodes or the edges themselves, but we're looking at the structure of the edges, right? And even more with this homological scaffold, with these networks, subnetworks that I presented before, they look at the contribution of each one of the edges, because you cannot filtrate the edges alone, because when the edge is born, the edge stays there. But you can actually measure the contribution to the edge that's been to the edge at each one of the holes that are dependent on this edge. Hopefully I did not mess up myself in this sentence. So again, they look at cycles or 1D holes, and they find that, hopefully, the number of holes in both cases is more or less similar, but the holes in the psilocybin is much more persistent. So you have much more holes in the network. So you have to imagine that the network has much more cycles. Again, we are trying to interpret this thing, so hopefully you are with me still, that the information from one point to another has to, yeah, at some point, right? But that's not so easy. That is not so easy. Then you look at this homological scaffold, and you see that you have some sort of, not contradiction, but very subtle difference. When you look at the global network structure, the persistence of long-range connections, because this is where it gets tricky, and I have a lot of trouble with this, but hopefully I can explain it. What does it mean that we have the same number of holes, but the persistence of them is different? That means that there are some holes that are closed rapidly, because that's what we saw in the first picture, but somehow they keep another one open. That's the only way I understood that this can happen. Because how can you close a hole super fast, but still be very persistent, right? I don't know if I should go back, but maybe this is, so if you look at the persistence of the psilocybin holes, it's lower, like it's clearly lower, yeah.
If I understand this correctly, maybe I got lost at some point. That could be a result of being the ways of the graph being more scattered, like more distributed.
Yeah.
Instead of
Yeah.
Can you repeat the question?
So the question was that this, at first glance, contradictory result, if it could be explained by the weights of the original network being much more scattered. Yes, I arrived at the same conclusion, and they said the same conclusion, and I read it like 2 minutes ago. But yeah, it's true. That's the only way you can imagine, because if we go to this example that I tried to illustrate, a normal network would be that perhaps the next connection would appear between this one and this one, so you would keep one community here, but you would not be closing kind of any hole in a way. But you have to add connections that contribute to the same number of holes, but holes that are in a way bigger maybe, so they can persist a lot more. So maybe that's the only way of doing that, is to build not only scattered networks, but to build long-range connections, so new holes appear, right? Because you don't want to, but still, yeah, but still holes easy enough to close so that the persistence is lower. Hopefully this kind of makes sense. This is the last slide, so you will be able to breathe soon enough. What they do is, okay, let's plot the other networks that they build that measure how many holes do edges participate and the persistence of those holes, and they find like this is astonishingly different actually. And what they find is that long-range connections are much more present and much more important in the psilocybin than in the placebo, which is something that I believe it's known and causes the network to be much more integrated and not segregated. Segregated means communities, so information to flow from one community to the other needs to go through the hub, through the connector hub. But that is not what happens in the psilocybin, where everything becomes much more integrated, so information gets also mixed up in the brain. That's why you hallucinate. I believe my understanding of hallucinations is that you have these two regions that usually operate separately, but then they are connected. So it's like I'm seeing, I remembered the cow yesterday. I saw a cow yesterday and I'm putting it now that I'm eating some pasta. So, but because it's information that it should be stored and kept separately, but now it has an easier way to flow, because these long-range connections appear in the psilocybin, which they see. I don't think it's a particularly groundbreaking study in the terms of understanding psychedelics, or psilocybin in this case, but it's a new way of looking at what we already know, that it's network much more integrated and with new long-range connections. So maybe this can be taken as a validation study, like, hey, we did this thing and we found what we already know. So can we actually use this thing to find something that we don't know? And since this is in 2014, like, this has actually gained a lot of momentum, quite a lot of momentum. It's still not in the level of connectomics per se, but so maybe there is a way to move forward connectomics with these specific tools of topology. Now, two questions for me that I wrote here is that this does not explain how the information is flowing in the network. We can only hypothesize about with the structure, but because functional connectivity is not effective, so again, this is not probably a problem of this method. And the other one, to me, much more puzzling, is that what does it mean exactly to filtrate the weights? I don't have an answer to that, but hopefully I've explained this thing in a much more clear way, maybe some of you has an idea, because when you compute your functional network or your structural network in this case, the network is what the method has done, like what the MRI has seen and what the correlations are, and that's the network. And they say, okay, let's like change the network and look at the topology of the network across these changes. But what does it actually mean? to change the network in a way. Like, are we analyzing something that it's fake, or are we analyzing something that it's integral of the structure and somehow has some kind of sense? Now, of course, they argue that all these filtrations are part of the network itself, but can we actually interpret this in a way of psychological? Like, what if those if those edges were not there? Of course, the topology would change, but what are we interested in the whole network that it is, because our brain does not filtrate, or it does filtrate? Like, does it actually threshold connections at some point or not? So this is like, I don't know, have an answer, but to me it is puzzling, at least the concept of filtration in a biological sense. And the idea would be that we have some data from stroke and some data with glioma, and we try to use these tools to see if they show something that connectonics doesn't. So yeah, that would be it. Hopefully I did not bore you too much with this intricate but yeah, hopefully I convinced you. There are some libraries in Python that implement some of these concepts, not all of them, but some of these concepts. So you basically don't know how to do you don't need to know how to do an integral over a path to use these things, right? I don't think you need to use any integral over a path to use these things in any case. But yeah, so thanks for your attention, and that would be it from my side. And I see that I did not record. I did not record it. Yeah, if anyone has any questions.
Those circles, they show like some kind of traditional measure, or this whole measure?
So this is the network that was built with the weights that I defined, and then they do community detection on that network, on the control one, and these weights are the weights of that network. So this, for example, would be, I'm inventing it, some cortical region, and the size is the degree of that network, so how many connections it gets, and the weights are the weights that take into account the persistence of the holes. It's not the original. Yeah, that's exactly. Like, if you want to understand this, you really have to.
Number of connections between like one edge and the
No.
other edge. This
No.
is already illustration of this new metric.
Yes, yes. And the difference is that you have
The
a
line is the persistence of the hole.
yeah, of the holes that an edge contributes. Exactly. It's very complicated to sell these things.
The hole that those two things like create.
Yeah, the edge. So
Those two edges create.
So yes, so we have the real connection. This means that, for example, these two, the connection is real. It exists there in the original network. But the weight is given by the contribution. So to how many holes does this edge participate? It's tricky, yes, and it's even more tricky. Like, what is this information? So what is this new network giving me more than the original, right? Just that it contributes to more holes?
Yeah, because I imagine you can do the same circles using traditional measures.
Yes. Now the connections will be different for sure. So this network is it's clearly not the same. If you binarize the network, you will get the same one, but the weights are different. Right? And they say that this elucidates that you get much more long-range connections, but it's tricky. I believe that if they did that without knowing anything about psilocybin, they would struggle much more. So because Robert knows a lot of psychedelics, so I believe that he knows how the networks change when you have psychedelics. But if they didn't know that, for example, I did this, I would struggle a lot to reach the same conclusion. In fact, what they claim is that there is some structure present and that is holding in the network of the patients with injected with psilocybin. It's just not randomized. They say that it has some structure that allows these long-range connections, but it's very tricky. I agree, it's very tricky. Yeah. No questions?
I have more.
Yeah.
And it's like the images that it's calculated on are what, T1? No. Ones?
An fMRI, bold sequences.
Bold.
Yeah, hemodynamic. No, yeah.
Yeah.
Hemodynamic.
Okay.
Blood, oxygen,
Not
yeah.
structural, it's functional.
No, functional. The problem with functional, there was structural, we have structural, but Giovanni mentioned that the problem with structural is that the network is less dynamic. So you have
Yeah,
like,
of course.
it's
Yeah,
like,
that's why I asked.
so there are some studies with structural and these things, but probably they are less rich, unless you do some kind of, yeah.
Okay.
Yeah.
Thank you.
So can we say then homological whole equals functional path or something like that?
Functional path?
Path connection?
No.
No?
No.
Not this way.
No, but no, because I mean a functional connection is a direct link. A hole is like a circle, like that goes, it's like a path that goes around the network and like goes back to the same dot without any connection in between. So you would need to do a cycle to receive the information that you sent. You cannot like back and forth.
Yeah, but then how to ask it? If the bigger hole, the bigger the cycle is,
Mhm.
and the
Mhm.
functional cycle is the brain.
Yes, it's a functional. Yes, I understand. Yeah, you can say it's a functional cycle because it's like a cyclic set of functional connections. Yeah. Again, functional networks are pretty static also. So I'm curious to see if you're aware of dynamic brain states, like instead of analyzing the whole fMRI experiment alone, you kind of do the functional connectivity first 5 seconds, second 5 seconds, so you kind of have brain states. I would be curious to do these things to brain states, like what is happening. Maybe this is an answer as to, at some point, some weak connection is really strong in the next brain state. But the average makes it kind of weak. I'm not sure if, but so I would be curious to do these things with brain states.
And then giving psychoactive, how to put it, the drugs, while there is this experiment, but don't tell the patient when.
Yeah, some placebo. Well, it's placebo. I mean, it's not control. It's placebo.
But then you have the same brain, exactly. Same brain.
If you can recover. Yeah, that is actually true. Yeah. It's cool. It's a good idea, yeah. I don't have data with psilocybin.
Some of them, it's like you adjust it through veins, so they don't know.
Yeah.
Some of them were waiting until you get.
Yeah. I would be quite frustrated, honestly, if I would be the placebo.
So at 2-1, I think it's very hard to do these things with psychedelic and putting people in an fMRI machine. Some probably will be already picking out.
Well, probably controlled doses, like it is functional.
Even if it's small,
You can
small psychedelic and put someone in a machine, an MRI. Imagine this guy.
you can combine it with something that is calming, for example, ketamine.
Ketamine. Maybe with ketamine
Psychedelics
makes more
plus
sense.
ketamine, so
would
you
make more
have a
sense.
psychedelic experience, but you may be calmer.
Maybe the conditions to participate in the study were
They properly select. People
maybe you
who
would
are
be
not
you would
picking out.
you would need to be, let's say,
No, instead, I had a more serious question actually than this.
I don't know, maybe I should read. Yeah, I just take that it's fMRI.
So essentially, the psychedelic are like serotonin-like receptor. So you add more of this to the brain,
Yeah.
and it's normal. You have more functional connectivity.
Yeah,
Plus
yeah.
you have this.
Yeah.
So, but I was wondering, so essentially, you increase the functional connectivity, and now you have more increased measure. So, are they then not redundant with the functional connectivity? So, do we need this, or this is just a validation of the increase of functional connectivity?
That's what I said. Like
So
that's what I
this
said.
thing
Like
is
with
still
it,
not clear if it's useful.
it is not wrong.
No, but it's decoration of increased functional connectivity.
It is not wrong, I guess. Yes. In this case, it is not wrong. But can we actually go further That's why I said, can this show something that cannot be seen with clustering coefficient? That's why I complained. Well, like mentioned, like is this just a fancy new thing, or yeah?
Do you have any idea from the brain perspective why combining psychedelics with dissociatives makes psychedelic experience a lot stronger?
Dissociatives? What do you mean?
Dissociatives, for example, ketamine or nitrous oxide, when you combine a psychedelic with nitrous oxide, you will have, for the time of the nitrous oxide action, you will have a lot stronger psychedelic experience. Dissociatives so antagonists of NMDA receptor. So why like switching down the brain, switching down activity of the brain using dissociatives would make psychedelic experience stronger if it is correlated with the higher brain activity? Sometimes it depends on a study. Some study shows not higher activity but lower on psychedelic.
I don't know. This is a very difficult question. This is a very difficult question. I don't know if anyone has an answer to that, but I can hypothesize. If you're fine with that. Maybe I say like something that it's crazy, but okay, so yeah.
But you say that they were finding more long-range connections,
Mm-hmm.
but the error characteristic was the same, was constant.
No, it can be constant.
It's different.
Yeah. Well, they don't look at that, but it can be constant for sure.
I mean, among across the persistence homology in the...
No, they do not compute that, because to compute the Euler characteristic, you need 0, so you need the vertices, then you need 1D holes, 2D, 3D. But I that's what, well, you know the paper, that they do it, but you need like a whole sequence of holes. And it will not be the same, I'm pretty sure.
Yeah,
That it will be different, yeah. Yeah.
Different.
Yes, I still haven't answered your question though, but go ahead.
Yeah, I have another question. And, because I'm interested, because some studies state that psychedelics make the activity of the brain higher, and there are also studies that state that they make the activity of the brain lower. So, like, what's the consensus when it comes to...
So the overall functional connectivity is higher, but like in stuff like the default mode network is lower.
Yeah, in default
If you
mode.
are looking at the default mode network is lower, but the others are higher.
Did you understand?
Yeah, I understand. So cortical activities.
Yeah,
higher,
cortical or except the normal.
So the brain is more active in general.
But in the default mode is less, which is
You're basically altering the functional organization of the brain. So I guess that it depends.
Actually, I'm thinking this now. Yes.
You
I'm
alter.
realizing it now.
Yeah. Yeah. So maybe depend on where you look, you find high or low. But yeah, the thing with psychedelics is that there's not really a consensus on anything, I think. Or in much more things there is not a consensus that in what there is a consensus. I don't know. I'm not an expert in psychedelics though. But you have to imagine that when you inject this serotonin, these are neurotransmitters. So you're essentially changing how the two neurons are connected. So if you put much more neurotransmitters, then neurons are much more connected between them. So like yeah, that's why you have much more activity. Let's say that a regular neuron needs 3 molecules of serotonin to get activated from the presynaptic neuron. If you inject much more, you will get much more serotonin, so you will get much more activity, which will be seen in the bold signal. Yeah. So if you, to try to answer your question, if you do this, maybe some neurons are sensitive to this serotonin, so you get much more activity, but if you add an antagonist, you will only antagonize specific neurons that are weaker, for example. So you somehow stimulate only a sub-circuit of your brain, hence altering even more the structure. So why do you hallucinate, or why do you have these things? Because the functional structure of the brain has changed. If you add an antagonist, you're only antagonizing certain parts of the brain, so even altering more, because I'm not sure if you're following my thought.
Because when one neuron is antagonized, this neurotransmitter will not be used to this unnecessary neuron and go to the other.
So I don't know. You have this is a brain, the brain of a mouse, and you add serotonin or psilocybin, and for some reason only these are affected by the psilocybin, while all of these remain somehow neutral. Like, so you have some sort of healthy structure still. But if you add an antagonizer, maybe you will not disrupt these excited connections, but you will disrupt the regular ones. So even adding a plus effect to the network. I don't know if this makes sense.
Yeah, but why would disrupting those neutral structures will add this plus effect?
Because this is somehow keeping a reminder of, hey, don't go nuts. This is what it should be.
Ah, okay.
But then you are like, instead of antagonizing this, you are like antagonizing this too, for example. So this is like not the brain of a mouse, but the brain of a cat, for example, now, in terms of structure, of functional. Does this make sense?
Yeah. Okay.
Is this correct? No, that it makes sense is not the same that is correct, right?
Yeah, I guess it wasn't like studied very specifically.
Well, I mean, if you add serotonin, not all the neurons will get excited by serotonin, and then if you add ketamine, not the same neurons will get antagonized. You know what I mean?
Yeah.
Like what I would find weird is that you add the same, so some stimulator and some antagonizer that acts in the same neurons. Then it would be weird to have like a plus effect, because that means that it's not antagonizing the added effect. I'm not sure if I'm explaining myself.
No, this.
So if you add some sort of a stimulate, so you add this thing, the serotonin, for example, or the psilocybin, and then you add an antagonizer that acts here as well, not here, but here, then it would be weird to have to have a more. It would be weird for me in this scenario, but if you're adding one random antagonizer, maybe it's like the psilocybin is altering, let's say, some cortical region, and the antagonizing is like silencing the hippocampus. So it's like that's even altering more. You know what I mean?
Yes.
Okay. But I'm not an expert, so don't take this for granted.
Don't make it a problem.
Yeah. No, don't cite this. Yeah. Anyway, that was it. Yeah. Yeah.
Okay. More questions? Well then.